All right. Good evening, everyone. So in this particular session, in just 30 minutes, I will explain you what exactly you people has to do for this 75 days. When our exam will happen, we also know very well our exam, see, they have said that either April or May, but most probably not most probably, mostly the exam won't happen in the month of April. Mostly it will happen in the month of May only. So we will keep that from today onwards, from yesterday onwards, if you count, or I'm sorry, from today onwards, if you count till April 30th, we have easily 75 days are there. If every one of you put little efforts, I mean to say, if every one of you put minimum of 6 to 8 hours of a dedicated preparation, it is extremely easy to crack the exam. Now, first and the most important point that every aspirant has to remember, whether you belongs to the general category, whether you are belongs to the BC, ST, SC, that is a secondary thing. I want you people to understand I want you to understand one very important point is that what is the safe score? It's a very important point. What will be the safe score in order to get the job? When I use the word safe score, when you reach this particular safe score, I can give you the assurance that 95 to 98% there is a possibility that you can get the job. Okay. Anyway, that you have to prove yourself in the interview as well, which is a minimal interview only. Now remember, out of 300 marks, I will always tell to everyone by keeping the general category person in mind. I want you to keep, and I want you to be in the range of 190 to 220 marks out of 300. And I told you minimum of 190. I don't encourage any one of you not to score less than 190. Never forget this. And what about general paper or the paper one? At least you should be in the range of minimum 60 and maximum 90. So if you are in this particular range, you will be around, 280 marks you are getting. So friends, if you are getting 280 marks out of 450, I can tell that is a kind of a safe score because we really don't know how exactly the question paper gonna be, and we really don't know the difficulty level. If you look into our 2018 paper, it is a completely moderate level. It is not very easy also, it's not extremely difficult also. But since the years are past, we really can't predict that how the questions can come. But you keep the point in your mind, 280 to 300 is the idealized marks in order to crack the exam. All right. So many people will start doing the preparation without having a perfect strategy. If you don't have a perfect strategy, friends, there is no point of reading it. So that's why having the strategy is extremely important. What is the meaning of having the strategy? Understanding your syllabus, understanding what was given, understanding how to, how to correlate the topics, how many marks that we can expect. Now, if you look into our DL syllabus, we are having Unit 1, Unit 2 non-card date, Unit 3 card date, Unit 4 cell bio, Unit 5 genetics, 6 human physiology, evolution, developmental biology, histology, ecology, immunology. Now, here comes a question. How many units that we have to crack, we have to study in order to be in the range of 280 to 300 marks? Every one of you listen. In order to get 280 marks to 300, including paper 1 and paper 2, I am saying, how many units that we have to focus on? Every student, remember, out of 11 units, remember everyone, minimum 9 units. I always use the word minimum. Minimum 9 units. That 9 units you should read 100%. You should read 100%. And what about the another unit? At least a half unit I want you to read. Half. I am talking only half. At least 50% you should be there. So in a simple words, out of 11 units, friends, I want you people to read 9.5 units compulsory. 9.5 units compulsory you have to read. Then only you can crack the exam. Now, I never encourage any single student from the unit 4 till the unit 11. So remember, unit 4, 5, 6, 3 unit, 4th, 5th, 6th, 7th, 8th. These 8 units, from where to where? From unit 4 to 11. I don't encourage even a single student also to leave any unit in this. If you are missing this 8 units, you blindly believe in your mind, you can't crack the exam. That's the fixed point I can say to all of you. Now, the very important point. What should I do? 1 second, 1 second. I'm sorry. It is going so fast. 1 second, just one moment. Now, what about the unit 1, unit 2, unit 3, unit 3? Because you people commonly know that, friends, it's a general concept. There is a no point of asking a very difficult question here. They never ask any kind of a difficult question here. What they will ask is a very simple, straightforward questions only. Correct, na? So I want you people to understand that here most of the questions are exclusively from the example based. I think you are already reading it. So this, whether the faculty teaches, faculty don't teaches, it is not the matter for you. Because please mute your mic. Please mute your mic. Do not make any kind of a disturbances. Right. So now, whether the faculty teaches or not, you can easily understand this unit. So I don't bother about this unit. I want you to focus on non-card date, card date. In the non-card date and card date also, I don't encourage all of you to focus 100%. Why? Because you already read card dates and non-card dates multiple times, and you have taught to the students also multiple times. You are much experts in this. Now, what my suggestion to all of you is, in the non-card dates and card dates, you make the tables. Remember my statements very correctly. I am not here to do the syllabus analysis once again. I have already done the syllabus analysis. Now you remember, you keep the PPT, whatever the PPT that you are having, that is shared by the faculty, you make the charts. What kind of a charts that you can make is an example. You are reading a porifera. An example, let's consider you are reading a protozoa. Okay. In the protozoa, how you have to make the chart? Example, remember all the students. So first you start understanding the locomotion, and exactly try to write only 5 to 6 points about the locomotion, only the key points where the examiner can stress it. And suppose if it's a nutrition, the type of nutrition methods, reproduction methods, most importantly, diseases caused by them. I don't want you to put an extra effort, or I don't want you to read an extra information which the question never comes actually. The question never comes actually. So that's why you should be very careful. My suggestion is that in entire non-card data and in entire card data, including card data and non-card data, friends, total we have a 16 small subunits. Now, what my suggestion to all of you, you keep the PPT given by the faculty and make 16 charts, not the paragraphs. Okay. Don't make any paragraphs. You make a 16 charts till the end of the exam. You only focus on those 16 charts in the non-card dates and non-card dates. But when it comes to the cell biology, you only see this picture. In our zoology syllabus, what they have done, they have combined the cell biology and they have combined the molecular biology. And in the morning class also, I have told you, friends, very clearly, in the cell biology, remember, you give the highest importance to the structure. Listen, every student: structure, functions, enzymes involved, and the diseases associated with that organelle. Once again, remember: structure, function, enzymes, diseases associated with that specific organelle. And what are the major organelle that you people has to give the highest importance? Remember, first one is the plasma membrane, followed by the nucleus, followed by the mitochondria, then, friends, RER and SER. Then very important one is the cytoskeleton. Then the sixth one is the peroxisome. Seventh one is the lysosome. And eighth one is the Golgi complex. Friends, these 8 organelles, under any circumstances, you should not leave it. You can leave the cell wall, you can leave the vacuole, small organelles, you can leave it. But I am requesting every one of you focus on this 8 organelles without fail. If you are missing any one organelle, you are losing the information unnecessarily. So that's why focus on all these organelles and read it very correctly. Now, In this syllabus, what they have done, apart from the cell biology, they have included the genetics here itself. You can see that. Now, friends, you very important one. Apart from the cell organelles, you blindly believe in your mind, you are going to get one question compulsory from the organization of the genes, and you are going to get one question compulsory from the cell cycle regulation, which I am going to finish completely in 10 days. Now you remember, there is a no question paper without the question from cell cycle regulation. I used to observe that most of the people will just focus on the mitosis and meiosis. You people never focus on, you people never actually focus on the regulatory part. So you have to focus on different type of a CDKs, different type of a cyclins that are actually involved in the cell cycle regulation. You have to read all of them very depth, friends. Then only you can crack that exam, crack that question. Whenever operon we have studied, you all people won't feel difficulty also. Apart from the operon, the most important unique topic is heterochromatin and chromatin. I don't know if you look into the TSPSC DL zoology paper, everyone listen. If you look into the TSPSC zoology paper, we got two questions. I'm talking about the DL paper actually. We got two questions from the heterochromatin and euchromatin. You know what question they have asked? In the euchromatin condition, what is the, please mute your mics, do not make any kind of a disturbances. Please mute your mic, mute your audio as well as the video. Do not make any kind of a disturbance. Okay, now they have asked a question. In the euchromatin form, what is the status of the H3 protein? In the heterochromatin, what is the status of the H2 protein? I mean to say, in the H2 protein, what modifications occurred? Whether acetylation occur, methylation occur, phosphorylation occur, same thing. H3 in the case of a heterochromatin, whether it is acetylated, methylated, phosphorylated. These kind of questions were asked. It is actually the question of the CSIR net level. So that's why when you are reading the heterochromatin and euchromatin, it is not like that. You have to just read the gene chromosome organization. You have to read extreme depth of the chromatins. You have to focus on the telomere completely. You have to focus on the centromere completely. And one of the most important topic under any circumstances, you should not skip is the transposons. What is the transposons? What you have to understand? Transposons are nothing but, listen my statements correctly, you have to focus on different type of a transposons. You know very well, we have a DNA transposons, we have a RNA transposons also. Apart from this, you have to focus on the characteristic features of the operon. Characteristic features of the transposons. Then followed by the transposons present in the plant, transposons present in the yeast, transposons present in the animal. So most of the time in the transposon, there is a chance that you can get either experimental based question or the statement related question. So friends, this is what you people has to read in the chapter of cell biology I'm talking. But you only look into this, friends. Apart from this, see how cleverly they have done. They have given the complete recombinant DNA technology, transgenic and cloned animals. This is our DNA part. What about the remaining part? Remaining part, that is the replication, transcription, translation, regulation of gene expression. That is what completely molecular biology. I already told to all of you what exactly you have to read in the molecular biology, and we have completed even this. Now we have to do this recombinant DNA technology that we will do after the immunology get over, because it's a very small unit. We can finish it in 3 to 4 classes. From our DNA technology, undoubtedly, I can tell you, you get a minimum of one question and maximum of two questions. From the transgenic animals, undoubtedly, you can get a minimum of one question. Now, in the rDNA technology also, is it necessary that we have to focus on everything? No. In the rDNA technology also, you have to focus on majorly vectors involved, enzymes used in the rDNA technology and their functions. Most important, screening methods. Screening methods is the major concept where you can expect the question. Apart from this, you can also get the question from the cDNA library as well as the genomic library. Friends, apart from this, there is no possibility of asking the question. So understand that very clearly and only focus on that. And I am giving you my assurance to you. That's all. If you sincerely follow every single lesson, and if you read, I'm telling you, out of 150 questions coming in the exam, whether you believe my words or not, you can understand 135 questions very easily. You can able to answer. Out of that 135, even though you answer 100 questions very correctly, that gives the greatest impact. So I'm telling you, because every question comes from the applied zoology majorly. I'm not saying that the questions comes from chordates and non-chordates. Let's consider chordates 10, non-chordates 15. Let's consider. Remaining 130 questions mostly comes from the applied biology itself. So that's why because chordates, non-chordates you can read. But here, instead of reading, you should require the wonderful understanding of the concept. And anyway, we are going to read the genetics in the evening class. Keep it in your mind very clearly, friends. In the genetics, you can get minimum 10 questions. You can get a maximum of 15 questions. If you want to read genetics, you compulsory has to read the book of Russell. But now I am modifying the PPT and I will give it to you. Whatever the PPT that I'm giving you modified PPT, it will help you 85 to 90%. And I have sent you hard copy of the books to the people who paid already fees. So that hard copy book, trust me, it will help you 100%. You no need to touch the book, any other source. And you ask me if the question goes beyond the level of that particular book. Every single question comes from there only. So you prepare according to that. What exactly you have to prepare in the genetics? Keep the point in your mind. In the genetics, just understand the normal principles, three principles of the Mendel, which is a common principles. After that, give the importance to monohybrid, dihybrid. And most of you people are leaving the trihybrid. You should not leave the trihybrid. Trihybrid is also very important. Apart from that, you should compulsory read the statistical rules while calculating the genetics, because from there only you get the numerical question. In TSPSC, JL also they have given that question. In the DL also they have given the question from the calculation part. So under any circumstances, you should not skip it. You have to read the multiplication rule also. You have to read the additive rule also compulsory. Then last time in the TSPSC, I have not explained the gene mapping because the question was not asked. So now anyway, we have to discuss that. We will majorly focus on linkage and incomplete linkage, which is very important. You gonna get the question. You can also get the question numerical from here itself, because linkage is one of the most important. And we will focus on single crossover. We will focus on the double crossing over also. That's a very important, friends. Then if you want to read the tetrad analysis, you read it. If you want to leave it, you leave it. But you majorly focus on the single crossing over and double crossing over. Then you keep it in your mind blindly, friends. You're gonna get two questions from mutations. You're gonna get two questions from chromosomal aberrations. You're gonna get 3 to 4 questions from the human genetics. I am talking about with respect to the diseases. I will tell you, whenever we are reading the topic, I will tell you what you have to remember for your exam. You only focus on that instead of remembering everything. In the mutation, every student remember what kind of questions we can expect. Tell me. You can expect a diagrammatic based question. You can expect a disease related question, right? You can expect experimental based question also. You understand? Suppose they will give one nucleotide sequence. Every student understand. They will give one nucleotide sequence and there was a mutation occur. They will ask what kind of mutation it is. That is important. Is it, I mean to say, loss of function mutation? Is it a gain of a function mutation? Is it a lethal mutation? Correct, na? Is it a point mutation? So these kind of questions are very common in that. That's why you have to read the entire mutations topic without leaving it. And again, a very straightforward question, a topic, deletion, duplication, inversion, translocation. And your advantage is what? In the botany, they have never mentioned the diseases. They have said to read everything. Your advantage is that they have mentioned the diseases very clearly. Down syndrome, trisomy, Turner syndrome, Klinefelter syndrome, hermaphroditism. Now, what is the advantage? You make a small chart here also. Make a small chart here also and understand. Remember my words and understand. Down syndrome is caused by deletion of which chromosome? Turner syndrome is caused by what? Klinefelter syndrome is caused by what? What I am trying to explain to all the students. For all these diseases, my friends, just remember their genotype. That's all. Just remember their genotype. That's all. Including phenylketonuria, alkaptonuria, sickle cell anemia. There are 80 diseases are there. For 80 diseases, you remember the genotype. Trust me, you're gonna get the 2 questions. And hardly it will take 1 hour. To remember also, it will take 1 hour. You make a small chart and keep looking into it. Don't read it. If you read it within 24 hours, you will remember. You will forget, friends. You just focus on looking into their genotypes and try to recap in your mind. Only these things you have to read it. Okay. It seems to be very lengthy unit, but genetics specialty is conceptual. If you know the concept, questions are in your mind. You can answer the question very easily. In your entire syllabus of DL Zoology and JL Zoology, entire syllabus, the biggest unit is the human physiology. Now, because here again, what they did, you tell me, in the human physiology, they have included a complete biochemistry. For if you look into the botany people, they have given glycolysis, Krebs cycle, PPP pathway, etc. And for the botany people, they have just given the enzymes. That's all. But for you, you look into your syllabus. First, they have given this is the exact syllabus of CSR again. Chemical bond and their interaction they have asked. Now you see this, but again they mentioned us outline the classification. Hence, you no need to read extreme depth. But whatever the PPT that I will provide you, that is 100% sufficient. Not even single question also won't come beyond that. Suppose if you're reading the carbohydrates, focus on monosaccharide, disaccharide, polysaccharide, especially their linkages. Whenever you are reading the polysaccharides, everyone will read only cellulose starch. That's all. No, it's not like that. You have to read glycogen, inulin, chitin, right? Glycopolysaccharides or glycolipids, heparin, chondroitin sulfate. Almost all the polysaccharides you have to cover. Then when we are reading the protein, focus on primary structure, secondary structure, tertiary structure, quaternary structure. In every single level, what kind of forces are involved? Suppose in the tertiary structure of a protein, which is the stable interaction, they can also ask the question. Every student answer my question. Among the primary, secondary, and tertiary and quaternary structures, which structure is the most stable structure? Imagine this is the question which will ask in your DL. Every student answer. Every one of you answer. Which structure of the protein, which structure of the protein is most stable? Everyone, you should not take this much of time. It is the 11th standard question. Sankirtana, Lalita, which structure of the protein is more stable? Radhika. Everyone, give me the answer. Don't be silent. I'll send all of you again to the 11th standard. How come you forgot? That's a small question, huh? The tertiary structure of the protein is the most stable structure, right? Because it is the three-dimensional structure, extremely biologically complex structure. That is only the most stable structure. How come you people forgot that simple question? Huh? And from the lipids, again, you have to read different type of lipids and the bonds involved also. Now, again, you zoology people, you have never read a concept called as Ramachandran plot. This is completely pure biochemistry. This also you have to read compulsory. Then along with that, the common thing, glycolysis, TCA cycle. Please mute your mics. Do not make any kind of a disturbance as I am telling you continuously. Don't disturb. Glycolysis, TCA cycle, PPP pathway. See this, gluconeogenesis, glycogenolysis, all things you have to read. Compare it to the biochemistry part of the DL botany, DL zoology is very high. That's why I'm telling the human physiology chapter is the biggest chapter for you in your entire syllabus. Now, how many questions we can expect in the human physiology? You see this, in the human physiology, there are 10 units are there. I can, you blindly believe in your mind, out of 10 subunits, you gonna get the 10 subunits compulsory. And I don't want to give the super summary of what to study, what not to study right now. When I'm explaining, I will tell you crystal clearly what to study. But I'm telling you the order of a priority you have to focus on. Start with always endocrine system, followed by the respiratory system, followed by blood circulation and cardiovascular system, which is the third one, followed by excretory. Fifth one, you read the nervous system. Sixth one, you read the muscle system. Seventh one, you read the sense organs. Ninth one, you read the digestive system. Why did I say ninth one is the digestive system? Because compared to all these systems, digestive system is so easy. You never get even a single question also, which is a extremely difficult questions. So easy questions you will get it. But in these units, there is a possibility that friends, you can expect the question in a very difficult manner. I already did the analysis of this. But when we are doing every single unit, I will tell you what to focus on. But example, if you're reading an endocrine system, what kind of questions we will get in the exam? Name of the hormone, from which gland it is releasing, what precursor is involved in the synthesis of that particular hormone, functions of that hormone, diseases related to that hormone, the most important aspect, that is the regulation of that hormone. This is the only way, this is the only area where we are expecting the questions. No point of getting confused over there. Now, I don't encourage any student to leave the small topic in the plant physiology. See, there are 14 topics are there. Minimum, you have to read 12 topics. Maximum, you have to read 30 topics, which means you have to cover 90% of the entire unit. If you are skipping it, keep it in your mind, you are losing 5 questions. If you are losing 5 questions, imagine where your rank goes. So remember that. And again, to make us very simple, the zoology double evolution syllabus, they made it so clear. So clear. Theories of evolution, here you only focus on Lamarckism, focus on the Darwinism, focus on the natural selection. Trust me, you gonna get the question from isolation and speciation for very sure. Again, there is a no DL zoology paper, there is a no JL zoology paper without the question from the Hardy-Weinberg equilibrium. 99% of the time, you get the numerical question only. If you get the statement related question, that will be difficult, but the numerical question is so easy. Again, apart from the numerical question from the Hardy-Weinberg equilibrium, you can also get the question from gene pool, gene frequency, genetic drift. Don't think that you get a statement related. In your DL, mostly you will get the questions which are numerical related here. The statement related are evolution of man, geographical reals of the world. And you are going to get, why I'm stressing these topics, you should not miss. Geographical reals of the world, you will get the question, one question undoubtedly, damn sure. Evolution of the man also will get compulsory. They will ask the question, what is the differences between the Australopithecines and Africanus? They will ask the question, the differences between the Homo habilis and the modern human. Otherwise, they will ask the question, arrange the following evolution of the man according to their cranial capacity based on the ascending order or on the descending order. That question also can come in the exam with respect to the cranial capacity. That's why we have to read it. But it's not difficult unit. So small, so simple unit. And I believe you all people read very well the developmental biology and zoology part. You are very good in spermatogenesis. You are very good in oogenesis. But I want, see this, they have just given a single line here. Fertilization, cleavage, and gastrulation. That's all they have given. But how big they are? You have to read the cleavage in the frog or amphibians. You have to read the cleavage in the fishes. You have to read the cleavage in the birds. You have to read the cleavage in the mammals. Same thing with the gastrulation. If you get a toughest question, you're gonna get from the gastrulation. toughest question. If not from that, you will get the toughest question from the development of frog and chick. Especially you get the question from the chick. Okay. In the chick, there is a concept called as a Henson's node. This is also one of the most popularized concept they use it to rise every single time. Then types of placenta, formation of functional fetal membrane. It's a very small concept only. You may get one or two questions. You people in the chapter of developmental biology, you can't read the entire Gilbert book. It will take a lot of time. Whatever the hard copy that I sent to you people specially, that only you read it along with PPT. I am giving you 2000% guarantee. Not even a single question also won't come beyond that level. So, read that very thoroughly. Then the madam will cover only 3 units, that is chordates, non-chordates, as well as histology. Completely she will only cover, and remaining else everything I will cover. Okay, everything. Okay, you keep it in your mind. April 26th or 27th is the last date. I will complete the entire syllabus and we will do the question solving. Now I will tell you. Now, when madam will explain the entire histology because it's a small unit, I don't want to spend much time on it. She will cover it. Ecology and immunology. Now, again, after the human physiology, after human physiology, the toughest unit is immunology. Why? Immunology is toughest unit. It is not because of the syllabus. It is not, it is because of the level of question that they ask in the exam. Not even a single question also you don't get very easily. I have seen the JL paper of the TSPSC when we see, and it is so easy, damn easy, I can say that. Everything JL they have asked just neat level. But when we look into the DL paper, in the immunology, everything they have asked about the net level. An example, what you have to read in the immunology very specifically. You just start reading from innate immune system, adaptive immune system, the differences between the innate immune system and adaptive immune system, in which immune system B lymphocyte and T lymphocytes will be present, what is the or which immune system will respond first to the damage, which immune system will show the long-term effect on the damaged area. These questions are very common and you should compulsory know the number, structure, composition, and functions of neutrophils, eosinophils, basophils, B lymphocytes, T lymphocytes, plasma cell, memory cell, T helper cell, T cytotoxic cell, T suppressor cell, natural killer cell, macrophages, dendritic cell. You just keep the point in your mind, whatever the cells that I have told you right now, all these 10 cells, you're gonna get minimum 3 questions exclusively from the cells of the immune system only. You should know what is their number, what is their structure, what they composed of, what are the specific functions performed by them. If you miss any aspect, you cannot connect this. So then after that, first is what? First you have to understand innate immune system, adaptive immune system. Then you have to understand cells of immunology or immune system. Without cells, you can't read anything. Then immediately you have to understand antigens. When you are understanding the antigens, you have to focus on the differences between the haptens and adjuvants, which is very, very, very important. And the most unique concept is the receptors. The receptors associated with the antigenicity, which is a very unique concept, very tougher concept. You have to remember it. And again, the important concept where there is no question paper without it, that is the MHC molecule, major histocompatibility protein. Again, their structure, composition, functions. MHC class 1, MHC class 2, MHC class 3. With the class 1, which will attack, I mean to say, T helper cell connected with which type of class 1, which type of MHC, cytotoxic T cell will be connected to the which type of MHC. This is also a straightforward question in the exam. Then, very important one, the 5 antibodies. 5 antibodies. Again, what? Structure, function, and in the DL TSPSC paper, they asked the question even from the molecular weight also. They asked a question, which of the following antibody having the highest molecular weight? Tell me, Sankirtana, which antibody having high molecular weight? Radhika, Lalita, Dharma, Anjanelu, which antibody having the highest molecular weight? It is also an intermediate question. There is no point of thinking, right? Can you hear me or not? Everyone. Okay. Answer the question, which antibody is in the pentameric in nature? Lalita, which antibody is the pentameric in nature? Every one of you. Which antibody is in pentameric in nature? Which is having 5 subunits? Radhika, why people are not responding? Am I teaching to wall? Come on, respond. Is there any problem in the audio otherwise? Why you can't respond to me? Which antibody having the highest molecular weight? Then why you people are not giving the answers? What is there in that? IgM. Why IgM having the highest molecular weight? IgM having the highest molecular weight because it is a pentameric in nature. Because they are having 5 subunits, right? So whichever having the 5 subunits, obviously it will show the high molecular weight. Anyway, you have to read structure, function, molecular weight, and the interactions between the antibodies. That's a very, very important. Then the important terms such as affinity, avidity. Friends, there is an important concept. I don't know whether you read it or not. In order to understand the antigen-antibody interaction, we will use a method called as a scattered equation. The scattered equation also extremely important for the exam. You can expect that also in the exam if the question is very tough. Apart from that, we have to focus on the diseases that they have given. Diseases, so that they are very easy. We can finish it in 2 classes only, so easy. But I can tell you, in an entire syllabus, the toughest unit is the human physiology and biochemistry. Then the second toughest unit is the immunology. The third toughest unit is the molecular biology and genetics. Fourth toughest unit is the cell biology. Fifth is the ecology. Sixth, developmental biology. Seven, evolution. Then chordates, non-chordates, and general concepts. So again, I'm telling you, whomever are there studying with us, you keep it in your mind. Keep it in your mind. Out of 150 questions that are coming in the exam, it is very important. We should be in a position to answer 110 questions. I'm talking least. Okay. I always talk to the least here. Least I'm talking. 110 questions, you should be in a position to answer it correctly. If you answer 110 questions correctly, trust me, you will be always selecting for the interview. There's no doubt about it. So keep the target of 110. If you are writing the TTD also, you remember this only. 110 is very important and mandatory. Now, friends, how many number of hours we have to read for this? Anyway, I will share this PPT with you. I have mentioned everything elaboratively from every chapter, what exactly you have to read it. And now, friends, how many hours you have to read? Okay. Now, if you start from today, that is 10th, 30th of the April, we are having nearly 75 days in our hand. So what I kept here is different. Immunology, I kept 10 days because this is the toughest unit. Developmental biology, one time revision, it is enough, 7 days. Genetics, conceptual. So some people, even though you read genetics 10 times, molecular biology 10 times, you can't understand it until you can't solve the question until you have the concept in your mind. So that's why never by heart. Genetics, biochem, cell biology, and molecular biology, don't try to by heart. You try to watch more number of times the videos until you have the concept in your mind. Then the first 3 units you give 15 to 20 days. And for the human physiology alone, you give 10 to 15 days. Because they are very crucial units, which are highest scoring units. How many hours you have to read? I know very well some people are already working, some people are free, some people are housewives, housewife plus working someone. But trust me, how difficult the situation it may be. Out of 75 days, if you don't read, if you don't read minimum 6 to 8 hours, trust me, there is no use of your preparation. Don't think that I already read the entire syllabus. If I revise last 30 days, I can easily crack the exam. That never happens, my friends. Never. You have to revise every single concept multiple times. So 6 to 8 hours is required. If I am taking 3 hours of class every single day, at least 4 to 5 hours of preparation is required, which is the self preparation. Now, when it comes to the question solving, now you people, I know some people are good in concept, some people are losing the confidence while solving the question. So you no need to worry. From the March 1st till the April 30th, I will start a session called as only question discussion. Okay. We have a 60 days of time. In the 60 days, I will I will just solve only minimum of 20 hard questions or maximum of 25. There itself, I will only do it. So I mean to say, madam will do for 3 units, remaining all units, I will do it. We will keep the target of solving minimum 1300 to 1500 questions in the live classes itself. That I will take the initiation, but you people also has to practice maximum number of questions from your side as well. Okay. Keep this particular timetable with you and strictly follow it. Now remember, whatever the hard copy books that I have sent, they are 200% sufficient. So my point is very clear. Keep the PPT and read the material only once, and whatever the questions I will solve, you people practice from your own as well. That's all. Don't do anything else. Don't read multiple books. Don't waste your time. Don't change your mind. Read the PPT and read the hard copy of the book whichever I have sent, and follow the MCQs which I will solve from the March 1st. You don't need to do anything extra, but whatever you're reading, try to become expert in that only. All right. Okay. Is it clear? Anyone wants to ask any kind of a doubt? Please raise it. Please raise it. Okay. I will keep the target of maximum of April 26th for an effective revision. So I mean to say for completion of syllabus. But we really don't know now whether the exam will happen on the right date or not. In Telangana, what they told that they have given the notification in the month of December. They told that exam will be in the month of April. They have given the notification in this one. They told exam will happen in April, but the exam happened in the nearly October. So, but we don't rely on that. If the exam get postponed, it's okay, fine for us. But we'll keep the target of April 26th or 27th. We will complete 100% of the syllabus. Then later, whenever the exam may come, you can do the perfect revision. Okay. And I'm assuring you, you follow PPT, follow the material and classes. Watch immunology class, human physiology class, biochemistry class, genetics class, developmental biology classes at least 2 to 3 times. Then only you become expert. If you just read one time, you never be expert. So that keep it in your mind and revise it. And keep the target that out of 150 questions, keep the target. I will always consider the general category student, and I'm telling 110 questions should be correct. This is the way you have to prepare for the exam. So prepare accordingly. Okay. Clear everyone? If you have any doubt, you raise it now. Clear everyone? See, Anjanelo, I completely understand. Not only you, even though general zoology students also will feel the molecular biology, genetics, biochemistry, little difficult only. What is the way? The only way is that we have to read multiple times. We have to read multiple times. See, if you read the replication only one time, you can't understand. You read it 2 to 3 times and apply your knowledge. So an example where the replication is taking place, what enzymes are involved, what rules it follow, how does it happen. If you remember the shortcuts, it is easy to remember. The only way is multiple revisions. Okay. And I'm telling to Sankirtana and Radhika and Lalita. I want you to answer all the questions. I don't know why you people are very silent. Huh? You are reading from very long time, still you people forgot simple questions. Spend more time on the preparation and keep reading it and keep revising it. No, Lalita. Actually, I don't want to take the headache of the paper one. Okay. See, now the point is that let me show to all of you. Okay. See, because whatever we do, we will tell that only. Why to take the another headache? See this. In this is the paper one syllabus. This is the only syllabus that we can do with you people, along with the CSIR. If you want to attend this logical reasoning, analytical ability, logical interpretation, you can attend. But remaining all, it is the general studies. It is not a mental ability. This is the mental ability, right? This we can help you. This is 9 and 10. Remaining or general studies, you find it something. Okay. Anjanelu, I completely understand, but you are not understanding a point that, see, I am teaching from the basics, but you don't know that I am teaching from the basics because you do not have any basics. See, if you are, 10th class child, if I keep it in a degree, okay, you feel completely difficult. That is what happening with you because you came from fishery to applied zoology completely. But you're not understanding. I'm always teaching from the basics. That's why for you, it's not like, misconception. The only thing is that you have to read 2 to 3 times. Then it will become clear. Once you read my lesson, read the material once, again listen the lesson. Then it will become easy. That is the only way. And the people who came from fisheries, you have to spend more time compared to other people. That you keep it in your mind. Okay. No problem. You can ask all the help whenever you want, but you put more efforts. All right. Okay. Thank you all. Thank you. I'll share the PPT, make use of it, and exactly whatever I have written, follow only those topics. Okay. Okay. Thank you all. Thank you. I'll see you sharp 6:30 p.m. in the class. Thank you. Thank you. Thank you.